And then our second speaker will be Karoline Maurer. She has a bachelor in biology from the University of Bonn and a master's in organismic biology, evolution, and paleobiology, also from Bonn. And she's currently a student at the DZNE, which is the German Center for Neurodegenerative Diseases, excuse me. She's also been a member of GHGA in the metadata workstream since May 2021.
Yeah, so also welcome from my side. In the second part, I will talk about the GHGA metadata model. And yeah, I will start with how we developed the metadata model. Then I will tell you about the trade-offs we faced along the way and how we dealt with them. I will give you in a nutshell, what does the model capture, what doesn't it capture, and why this is the case. Then I will talk about the metadata spreadsheet and show you the most important features. And in the end, I will give you a short outlook on what we planned for the upcoming GHGA phases. Yeah, so in April of 2021, we started off with an initial survey among the data hubs that are associated to GHGA, and we wanted to see how the local metadata requirements align with EGA. As you have heard in the introduction earlier, GHGA is a federated node of EGA, which means that we have to find a certain consensus with the EGA metadata model. Then in July of last year, we published our prototype schema version 0.01. And this was our prototype schema, which was largely inspired by EGA, but we added a few tweaks, which I will show you in a few slides. Then 1 year ago, we published version 0.4. Here we had extended our metadata schema, and we also introduced the submission spreadsheet, which the submitter can use to submit metadata to us. In the months after that, we did a lot of mock submissions and sanity checks, and then introduced version 0.6 in June of this year. And with version 0.6, we also introduced the transpiler service, and this offers automatic transformation from the spreadsheet format to a JSON format, and it also does baseline validation checks in principle to see if all the mandatory properties are submitted. In September of this year, we published version 0.9, and here we aligned the spreadsheet with our YAML source of truth model, and we introduced further fields based on feedback we received. And then right now we are in the middle of a major revision, and we are defining the controlled vocabularies and ontologies. And in the following minutes, I want to focus on our prototype schema, then how we extended the metadata schema, and then how we are defining our vocabularies. So here you can see the EGA schema on the left and our prototype schema on the right. And what you will also see is that the majority of the entities are shared between the two models. At this point, we did not adapt the assay and the analysis from EGA, but we added 3 more entities, which are the publication, the library, and the member of the data access committee. You can also see that we try to simplify the relationships between the different entities. And we now have a hierarchical model, which is dataset centric. After we knew what our prototype model looked like, we started to explore other databases and their metadata models to see what else is out there, what we can include into our model. And the databases we looked at were mainly focused on rare disease and cancer data sets, because these are also the communities that will be baseline communities within GHGA. So what we did here is that we looked at the models and the data dictionaries to see what properties they capture and which properties are shared among databases, which in turn means that these properties are important for the respective community. We also looked into minimal reporting standards, which are mainly focused on experimental properties. And here we looked into the minimum information about nucleotide sequencing experiments standard, which is MinSeq. Also the single cell equivalent, MinSeq Plus. We looked into the Genomic Standards Consortium, GA4GH, and of course, we are in alignment with the FAIR principles. We extended our model, included it into our prototype, and then we conducted another survey across GHGA stakeholders. And here we presented our extended model and asked for feedback and suggestions from the community that will in the end use the model. And all in all, this exercise was focused on the experimental and clinical properties and how they are linked in other databases. It allowed us to study the granularity at which properties are represented in other databases, and it allowed us to define what our model needs to deliver to the communities. So now we can have a look at the evolution of our model, the prototype on the left and our most recent model on the right. In comparison to our prototype, we now added the entities analysis, biospecimen, and project, and we redefined the hierarchy of our model. So now you can see that we have a study-centric model, which means that with each submission, the submitter can only submit one study. One study in turn is captures one or more data sets. One data set captures one or more experiments. Each experiment captures exactly one protocol and one library preparation protocol. And within the experiment, we have one or more samples, which are then, so each sample is tied to exactly one individual. We also introduced mandatory and optional entities, because not every submission will already have every entity that we capture. And these optional entities are the publication, the project, the analysis, and the biospecimen. So right now we are in the middle of defining the vocabulary we want to capture at GHGA. And as I said, we are a part of the federated EGA, which means EGA has already a list of terms that we can use for different properties. And for example, we can use lists that describe different entities, for example, the dataset type or the study type, but we can also use lists for the experimental properties such as library selection or layout. Right now we have the option to add terms, but once we have an exhaustive list, we will deprecate that option. Then we also manually defined controlled vocabularies. This was the case for properties where there is no ontology available, such as the instrument model, but also for others it was necessary because there may be ontologies, but they are too detailed for our use, for example, for the karyotype. And lastly, we also implemented ontologies. For example, the data use ontology. We use Brenda or Oberon for tissue and cell line, and for the phenotype and the disease diagnosis, we have implemented ICD-10 codes, the Genome-Phenotype ontology, experimental factor ontology, and the orphaned rare disease ontologies. While defining our vocabulary, we always were in discussion with experts from the respective fields to make sure that the terms we are defining and the ontologies we are choosing are community accepted. Along the way, we ran into different trade-offs we had to balance. And one of the biggest trade-offs was the data reuse versus the data protection. So of course, we want to provide detailed subject information, but we also have to be GDPR compliant. Our second trade-off was granularity versus excessive demand. We want to capture detailed metadata, but at which point does the detail become too much detail? Third, we want a simple model that is easily to understand and overseeable, but at the same time, studies are increasing in complexity and our model needs to be flexible enough to capture also complex use cases. The fourth trade-off is that we serve different communities, which also have different needs and expectations, and we have to find a balance between these two. So going into our first trade-off, we already meet our biggest challenge, and this is that metadata collected with our model will be publicly visible, and granular metadata might lead to data subject re-identification. So here we have to balance the data reuse on one side, which offers rich and granular metadata, which in turn makes data interoperable. And on the other side, we have the data protection, where we have to be GDPR compliant and allow the anonymization of data, which in turn protects personal and identifiable data. Together with Simon, our data protection coordinator, we found the solution to de-identify data subject information as much as possible by aggregating the information. This means that, for example, for the ages, we defined ranges of 5 years, which the submitter has to submit. And then you will also find this information aggregation at the diagnosis and phenotype properties, where we included a disclaimer that the submitter has to think about whether the term they are going to submit is so rare that in combination with other properties, this may lead to the data subject re-identification. For our next trade-off, the granularity versus too much detail, this is mainly focused on that we want to enable a proper search function, but at the same time, we want to avoid that a person is submitting data to us and while going through the catalog thinking, I have now answered the same question a million times now, why are you still asking this? As an example, for some experiments, it might be enough if we capture the sequencing model and the library kit, but as soon as some in-house protocol is used with a makeshift library kit, this is already not enough properties to capture the necessary information. On the other side, if we are too detailed, then a lot of the information might not add any real value for a data reuser, and maybe some of the properties can be recalculated by a data reuser. So we had to find the golden way in between these extremes. And our solution is that we avoid redundancy because we define a hierarchy at the experimental level, and we also infer properties based on other properties. And in accordance with the minimum information standards, only the most important and common properties are mandatory. This also means that there's always a minimal catalog that is browsable and slims down the search function. Then our third trade-off is simple design versus complex use cases. And here I want to go over 2 use cases we ran into quite often. And one of them is that we have one individual that has multiple samples. For example, if we have a cancer patient that has several tumors and each tumor is then sequenced. And here we take a look into the relationship between the sample biospecimen and individual entity. You can see that one individual can have 0 or more biospecimen, which would then be the different tumors, and each biospecimen has 1 or more samples, and the samples are then what is in the end sequenced. I told you in the beginning that the biospecimen is an optional entity, and this is why we also implemented the direct link between individual and sample. And individual and sample have a one-to-many relationship, meaning one individual can have 1 or more samples. So our use case with one individual, multiple samples works. Another use case is that we have multiple samples in one file. And here we take a look at the relationship between sample, experiment, and file. And here you can see that there is no direct link between sample and file, but the linkage happens within the experiment entity. So one experiment can have 1 or more samples, and one experiment has 1 or more files. This means in turn that sample and file have a many-to-many relationship, meaning one sample can have multiple files and one file can have multiple samples. In addition, we capture the barcode size, offset, and length in the sequencing protocol and the sample barcode sequence in the sample spreadsheet. And this is in accordance with the MinSeq Plus standard. Our fourth trade-off is that we have different communities with different expectations. And for example, the cancer community might need tumor-specific information, for example, the staging of the tumor and also the karyotyping of single cells. And in addition, the detailed exposure to risk factors. For the rare disease community, on the other side, they need pedigree information because they mainly deal with diseases that occur within the family. They need the karyotyping of single individuals instead of single cells, and they need detailed information about the phenotypic features. Our solutions here is that we included the most common characteristics between the two communities, and this was mainly a result of our database exploration exercise. We are in discussions with experts from both fields to see what their specific needs are and how we can implement them. And right now we have our core model going, but we still have to further look into how we can include more detailed clinical data. So in a nutshell, what does our model capture? If you look into our spreadsheet, you will find a lot of aliases, and these are mainly used for internal linkage. Some of them are also used to be shown as IDs in the catalog later on, but whenever this is the case, you will see a note below the alias. Our model captures detailed experimental metadata. This makes the data reproducible and allows users to search for a variety of experimental combinations. And our model captures aggregated information at the individual level, which enables the search for a phenotype or diagnosis category by preventing re-identification. And a note to this is that not for every individual the phenotype or disease has to be submitted at a higher level. This is just for single cases. So for most, this will not be a diagnosis category, but proper detail category. So then what does the model not capture? It doesn't capture detailed information about a data subject. And as I said, the metadata will be publicly visible. We don't take any liability, but we want to prevent data subject re-identification. And we also want to protect the sensitive data that is submitted to us. The model does also not capture every possible detail about the experiments and analysis you have performed. We are in accordance with minimum information about any sequences standard, which allow reproducibility while not frustrating users of GHGA during submission or the data search. And as I also said, we have, with our mandatory properties, always a minimal catalog that can be browsed. So you have heard about the FAIR principles, the latest 20 minutes ago in Anandi's talk, and I also want to quickly show you how we implement the FAIR principles, not only for the data that is submitted to us, but also for our metadata model. Yes, data in GHGA is findable because we ask for rich metadata and we have mandatory entities and properties. Data and the model are accessible. The model is readable by humans and machines because we use a YAML file format, and the data access is managed by a data access policy and a data access committee. The data is interoperable because we use controlled vocabularies and ontologies which are community accepted, and both the model and the data are reusable. We capture technical metadata. We are in accordance with minimum information reporting standards. And for our metadata model, we also have a GitHub repository where you can find the metadata schema. If you go to the repo and you find the link to the spreadsheet, yeah, as I said, I won't give you the big tour of the spreadsheet right now because we are still in the middle of a big revision, but I will show you the most important features. So what you will find in the spreadsheet is a sheet called submission information. And here you will find information about the metadata version, you will find contact information, and you will also find a list of our controlled vocabularies. These are sorted by property, and whenever we have an ontology implemented for a certain property, you will also find a link to the respective ontology. We also included a submission checklist, and this is meant as an overview, and the submitter can see in one sheet what entities we are capturing, what properties we are capturing, whether these entities and properties are mandatory, optional, or conditionally mandatory. And the submitter can also go back to the submission checklist once they filled out all the spreadsheets, and then check off the little boxes to see what is still missing or what else can be submitted. And lastly, we have a guided submission, which means that we arrange the single sheets in a bottom-up approach. So first, we capture the basic information about the study, then we register the files, tie them to an experiment, then the samples are registered, then if an analysis was performed, the analysis is registered, and then in the end, everything is tied to a data set. The link between dataset and study then happens internally. In the single sheets, you will also find disclosure considerations, which are always highlighted in red. And here we tell the submitter to please not make up any aliases based on personal information from a data subject. Then you will also find property descriptions for each property. We have a row with the data types we expect under a certain property, and we also included a short how-to. As I said, for the aliases, you will also always find whether this alias will be shown publicly. And for the properties where we have a controlled vocabulary, you will find a link to the submission information to see the list with the permissible values. Our future plans for the next GHGA phases include that we want to keep the model flexible to make the necessary adaptations as GHGA and the goals are evolving. GHGA wants to offer standardized analysis at one point, and we are thinking about how we can capture the processing metadata for these workflows. Two things that we want to capture are the quality control metrics and, for example, the workflow versions. And as I said, we are also thinking about how we can include the clinical metadata, and this is a process that is ongoing right now. And last, I want to take the opportunity because I'm the one presenting here, but this has been a team effort. And I want to thank everyone that was and is still involved in developing the metadata schema, and to everyone who gave feedback and suggestions. And yeah, this is the end of my talk. So thanks for listening. And if you have any questions, I'm happy to take them.